Well, I will start slowly because I can see that people are, first of all, my apologies for the terrible title. I don't really know whether Tom asked me for a title or a description of what I was going to talk about, but it is pretty self-explanatory-ish, but this title probably would be better. So we are going to be discussing, I will be presenting about something that we have touched upon quite a few times in the last 2 days, which is what can microbes do, or what do microbes do? It is something that it is the song, it's not the singer, etc. We have used different ways to refer to the microbial functionality, and I have added a few names here. Lone, Anna, Jacob, Jan, Asmus, Clarissa, Thibaut, and many others were talking about microbes as the functions and how important understanding those functions is to really get a grasp of how microbes can be affecting their host's biology. And let's start from the very, very basic. For those that are new to this, so what we basically do is whenever we are doing shotgun metagenomics, genome-resolved metagenomics, we try to generate genome sequences of bacteria that are as long as possible. If possible, they are circularized. And then in order to get functional information, we first need to predict the genes. We get based on gene prediction. And then we need to assign functions to these genes. And this is called gene annotation. So this gene annotation, explained in a simple way, what it is done basically is to match our gene sequences against gene sequences that exist in reference databases that have functional information annotated. Some of the most widely employed ones are KEGG, for instance, up here. This is the icon for case size, and case size are those GH case size families that Itzik was showing on the Rominococcus, etc. We also have the protein families, PFAMs, etc. So we have nowadays the capacity to use this ensemble annotation to altogether try to get as many annotations as we can for each of the genes that we predict from the bacterial genomes. And we do so not just for one genome at a time, but we can do that with hundreds or thousands of bacterial genomes. And that oftentimes yields this type of situation in which we might have thousands of genomes, thousands of bacterial genomes that might be more or less complete, which contain millions of genes. And this is usually the reaction of any student, any researcher, and say, yeah, I have millions of genes, I have a lot of information, but then we need to make sense to this. And usually when we try to make sense of this, it's so complex, and we always return to that concept that we introduced in the first day, complexity. The complexity is so big that we really struggle to make sense to all this ocean of information that we generate. Because usually, in our case at least, we work only with dozens or hundreds of samples. And we end up in a situation where we have the well-known big P, little n situation, and we have very small sample size, and then the parameter space we are trying to get some information from is immense. We have millions of genes, we have thousands of genomes, etc., and very little amount of samples, and that's a problem for statistics. That's a big problem for statistics. So one important thing that we do, and we have not really touched upon this that much in the conference, but I think it's really important so that we can make sense of this data, is dimensionality reduction. So there was a PCA shown before. There has been some different ways for doing this dimensionality reduction mentioned during the conference. And it is, I think, important to mention that these two ways to do this dimensionality reduction. One is the unsupervised approach, in which you rely on the data and see how the data clusters itself so that you can group together pieces of information that seem to be working together or behaving together. While we have then the supervised approach that is based on using the prior information we have on the system to create some kind of a structure on the data, and that way, rather than working on the raw, immensely large amount of data in clusters that are biologically relevant. So for the unsupervised approach, Juliaxis, for instance, yesterday introduced the enterosignatures. We have the enterotypes, etc. All these are based on the data, are based on coabundance matrices, co-occurrence matrices, etc. When we try to see how microbial species behave together, and based on that, we can start constructing microbial guilds, etc., etc. And that gives us some possibility to reduce from thousands of bacteria to perhaps dozens of guilds, and then we kind of reduce part of the problem by doing this dimensionality reduction. Another approach, which is not better or worse, is different, and they can enable us to do different stuff, is based on the actual information we know about bacteria themselves. And not just bacteria, but also the genes that they encode for, the functionalities of those genes, etc., etc. And I wanted to stress here this concept of hierarchy, because life is quite organized, and I think we can take advantage of this organization so that we can create nested structures to make the most of the data we generate. And today I will mainly focus on data we can generate and we can extract from these two main databases, which are the KEGG database and the Metapsych database, which do not only contain information of what genes do, but contain huge mazes of metabolic pathways that can interrelate all these genes. And we can start getting some more insights into how all these genes could be contributing to the biology of bacteria first, and then how the community functioning could be affecting the host. A way to use this information for dimensionality reduction is to rely on the pathways, on the modules that reduce the complexity and at the same time set some more context. Because sometimes just a gene ID and enzyme commission code, etc., might not be very understandable, but if we have a pathway that tells you, okay, this is the pathway so that you can biosynthesize vitamin 12, or you can degrade tyrosine, or any other things, that suddenly starts becoming something that is more palatable for regular biologists. And using this kind of information, we usually in our group use two alternative approaches, or two complementary approaches, because this information can be used in many different ways, and a lot of people use it in many different ways. But today I'm going to introduce these two approaches. The first one is based on the genome-inferred functional traits that we call them gifts. It's not something that we have invented, but we have named it here because it's cool to come up with an acronym that's GIFT, and in this context, and as we are in Denmark, and in Denmark acronyms are a big thing, everything has an acronym, so we came up with this GIFT, and hopefully it thanks. And then we have the genome-scale metabolic networks that I will also touch upon. And I will then, in the end, showing you that we have developed two R packages, actually, to work with one or the other of these options. And I will show some QR codes as well. So for the genome-inferred functional traits, we have this pathway, this imaginary pathway, and the idea is extremely simple. So the gift value will always range between 0 and 1. It's going to be 1 if all the genes for conducting this function are present. There might be some redundancy. This is also a gift 1, because not all these genes are really needed in order to conduct this process. But then, as we remove genes from the system, then the gift value will be reducing until we have a gift 0. So when we have no genes at all, then we'll say, okay, there is no potential at all. When we have a O33, that's something that is high. Should we consider that as present, some kind of evidence that there is this functional capacity? This is something that can be discussed for hours and hours. But at least we have a quantitative way to start reducing all this information into traits that can be then analyzed statistically. And this is what we get. So here in each column, we have a different MAC and a different genome, metagenome assembled genome, and each row here is a different gift. And using different databases, we can take all these millions of gene annotations and get into this kind of contingency tables that then we can far more easily input into the statistical modeling. This is something I think Thibaut also showed yesterday, an ordination of this functional traits. And here what you can see is that we have some Bacillota here that are, the blue ones are the Bacillota, Bacteriolota here in brown, and their position is based on their functional traits. So this is not the ones that are closer to each other will be functionally more similar, and we can see here how we have some Pseudomonadota down here and some other Pseudomonadota up there that are functionally radically different. So this is one possibility that we have this information, which is just to ordinate it and visualize it. By using this package we developed, the distiller, called distiller, what we can do is not just to distill all this information, but also organize it in a hierarchical way so that we can analyze the data in different levels of resolution. And this is quite important because depending on which statistical tools we are using, then we might be able to input 200 gifts, or we might not be able to use more than 8 or 10 because we might be relying on Bayesian methods with really long MC-MC chains that need to be converged, and then if we include too many parameters, then it won't converge at all, etc. So we developed this tool to create this scaffold of biologically meaningful attributes so that the functional information could be properly analyzed. And these are some examples. So we have this paper under review. It is very simple, but we just compared how the functional traits that we can recover from the fecal samples or the cloacal samples of these Celoporus lizards look like. And then we can do these kind of things where we have the different capacity for biosynthesizing the different compounds here clustered by the main functional groups, and we can display the differences between them. We can also do modeling on this, and this is a bit more advanced methods where we input this information into hierarchical modeling of species communities framework, in which we can then model how the community dynamics change, in this case, in an omnivorous Apodemus sylvaticus and in an insectivorous Crocodilura rusula. And this is an experiment that we ran a few years ago and was published the last year. But it is based also in this gift information that then we input into the modeling to not just get the community composition differences, but also how this functionality changes. And then we can scale this up to the really huge scale. And this is what we are doing also in the Earth Hologenome Initiative, where we have so far recovered over 60,000 bacteria from almost 250 vertebrate species. And we start creating this kind of maps where we can a lot of functional traits and then try to start seeing where the patterns exist based on what are the phylogenetic relationship between the hosts, what are the geographical relationships, their functional relationships, et cetera, et cetera. The other approach is to use the genome scale metabolic networks. And we can, starting from the same annotations, what we can do, rather than using each pathway as an isolated unit, because that creates some problems. This has other problems, but creating discrete packages, it's quite subjective. Where does a pathway start? Where does a pathway end? Because a lot of these pathways are interconnected. So using genome scale metabolic networks, we can create this kind of pretty maps. And I think Jan showed a little bit of this yesterday. And we developed this other package called MetaDeep to do some kind of statistics that existing packages, as far as we are aware, are unable to do. And the main difference here is that what we try to do is to see how the potential of different bacteria to interact with each other in terms of metabolite provision and usage is. So we defined source metabolites, which are the metabolites that a bacterium is unable to generate itself, but needs to get it from somewhere else. We have transit metabolites that are the ones that are in between, that can be used and can be produced. And then we have the sink metabolites. Those are the byproducts that a bacterium is unable to use, but it does produce based on its metabolic machinery. And then if we have all this information, we can start relating bacteria to each other. And if we know the relative abundances of each bacterium, then we can do some statistics and see what is the potential that each bacterium has, or a community has, to fit a specific bacterium. And this is how it looks like, the genome scale metabolic networks with the source, transit, and sink genes or metabolites shown here for 2 random genomes. And then if we put this into the context, then we can see in this case that that was an example by Sophia, in which, and this is an output from Holofood, the previous European project, in which we showed that the community development of one type compared to another type of community development was creating an environment that was more or less favorable for Campylobacter to colonize the intestinal environment of chickens. This information can also be used to create hypotheses. We're using genomic information, using genomic-scale metabolic networks. We can say, okay, what is the likelihood that we have a priori, based on all this modeling, for different bacteria to interact with each other? And this is where 3D omics, which has been introduced already by Anna, and mentioned by Itzik, and then we will hear more from Brian about it, comes into place. Because in 3D omics, we are also using this kind of extremely small sections of the intestine. This is a cecum of a chicken. This is 1 centimeter more or less, and each of these discs is extremely small. But now we have the potential to know how the community of the microbes changes from here to here, from here to here, and we're going to start actually testing whether the predictions we create from this metabolic modeling can be really observed in situ at the microscale, at the scale at which microbes interact with each other. So microbial functions can help us illuminate all these interactions, and if we merge them with other tools that we have, I think we can really do breakthrough discoveries in the near future. But, and these are just 3 of the limitations, the caveats that we have, we are always limited by existing annotations, and this has been mentioned a couple of times already in this conference, and I pulled some information from our data set on the Earth Hologenome Initiative, and this is out of the 60,000 genomes we have reconstructed, this is the percentage of unannotated genes. We can see sorted by the most unannotated, so it's 60% of the genes that have been predicted in this phylum do not have any annotation at all, and this is based on ensemble annotations using PFAMs, MERAPs, cases, KEGs, etc., etc. And if we reduce that to the KEG orthologs, then we see that on average, like 50% of the genes we annotate in these genomes don't really have a KEG annotation yet. So it is everything that I told you that could be done cannot be done, actually, or can be done only with half of the genes there, while having the other half that we have no idea what's going on. Another limitation is that we are constrained by the existing pathways, and this is quite important to remember, which is we have this pathway, for instance, for biosynthesizing L-ornithine for metapsych. It says that expected taxonomic range is archaea and bacteria, but the actual bacteria this pathway has been tested in is very limited. So we are extrapolating into a pathway that has been described, has been tested in 3, 4, 5 bacterium, and then extrapolating that to the immense diversity of bacteria that we have out there. So this is another thing that be cautious of what we do. And the last one is also the bias that we get from incomplete genomes, and this is something that we showed the last year in a publication that we put out, in which, just to summarize what we saw, is that if you have a Pseudomonas aeruginosa that is reconstructed at 70% of completeness, suddenly functionally it becomes a Bacillus subtilis. So basically, do not trust the completeness of bacteria reconstructions that have rather low completeness values, or try to at least account for that in the statistics, or at least measure what biases this can introduce. So, and these are just a few of the limitations. We have also heard, we have plasmids, we have phages, we have so many other things that are messing around everything. Well, at least we can get closer to answer this question and to say, well, to try closer on what microbes can do. And hopefully, this was a decent introduction of what are the potentials, but also what are the limitations that we nowadays have with all these functional inferences. So I am running out of time, so I would like to thank all the founding agencies that make our research possible, and I cannot be more grateful to the wonderful team that work together. Most of them are here, so kudos to all of you, because all this would be impossible without you. So I think we have 1 minute for a very fast question. So thank you so much.
We do have 1 minute for one question. Oh, there we go.
Thank you, Anton. So with regard to the gift, so let's say that you have 90% of a genomes in a specific pathway and you need one gene in order to make it work. How would you consider this? Like you need some context information with regard to the pathways themselves, no?
Exactly, yeah. It is something that so unless we generate enough information in situ, in experimental information to know how close we are to the target, I don't think we can make any sound decision on where to set the threshold. So that's why, as there is this potential for complementing each other, even though we don't know whether these metabolites will be released to the outer environment or not, oftentimes we don't have that information. Right now, the package contains the possibility of considering one binary options, where you set the threshold where you want, and above that threshold is 1, below that threshold is 0, or otherwise you take the actual proportional value that you want, because right now, I don't really dare to make a, a decision on what would be the best way to describe the biological reality. So.
Thank you, Antton. Sorry there was less time.